we have studied how the biotechnology is applied in agriculture. By the help of biotechnology, we can make insect-resistant plants, pest-resistant plants, genetically engineered plants. It's used for different varieties, different kinds of purpose. Today in this session, we are going to study how this biotechnology is applied in medicine. It's applied in medicine to make different medicines like antibiotics, vaccines. It's used to save different millions of people in everyday life. So first of all, we are going to study about insulin. As we know, insulin is a hormone. It's secreted by pancreas. One of the endocrine gland, it is pancreas. It is the insulin. It is used to control blood sugar level. If the blood sugar level is increased, then the insulin it is used to reduce. It's used to control the blood sugar level. Nowadays we can understand there are many millions of people they are suffering from diabetes. So these all people they need the insulin therapy. How can we make? So at first scientists they started to make insulin from salted cattle and some other animals. So slaughtered means because of this insulin they have to kill. There are many cattle. So and not only that insulin it's used in human beings. It causes different allergic reactions in the human body. So now the scientists they decided that how we can make insulin from human from the human body itself. We can make the insulin. So they started to study about the insulin. So first of all, let us study what's the structure of insulin. Insulin, it is made up of, it contains two polypeptide chain. Insulin is made up of two polypeptide chain, that is A and B. This A and B, it's linked with disulfide bonds. It's linked with disulfide bonds. Disulfide bonds. So this is A and B chain and it is linked with the disulfide bond. So this you can see the structure of insulin. But what happens when the insulin is secreted in the human body? It is like proinsulin. It is called proinsulin. Proinsulin. Already we have studied, we have understood like in Bacillus thuringiensis, it produced Bt toxin. It's produced Bt toxin. It is called protoxin. It is inactive in Bacillus thuringiensis. Same like here, the insulin, it is pro-insulin, it is inactive. So what happened? The pro-insulin, you can understand, in the pro-insulin, the polypeptide chain A and B, it contains the one more structure, it is called the C chain also will be there. And A and B it is connected with the disulfide bond, but in between C chain also will be there. That you can say proinsulin. The structure of proinsulin. So this proinsulin cannot act. This proinsulin has to convert it into mature insulin. It should be converted into mature insulin, then only it can act, it can control the blood sugar level. So you can understand like this, in mature insulin free of C peptide, but in proinsulin C peptide also will be there. So now one of the company name is Eli Lilly company. Eli Lilly company. This company is from America. So that scientists they have decided we can make insulin from the human body itself. What they have done? They have extracted the gene responsible for polypeptide chain A and B. What they have done? They have extracted polypeptide chain A and B. The gene responsible for polypeptide chain A and B they have extracted. And that gene they inserted into the bacteria. It is Escherichia coli. They have inserted. In the E. coli, they replicated, means large number of copies they made this polypeptide A and B chain they have made. Then what they have done? They have extracted all the copies of polypeptide A and B chain outside. Then outside they have linked this polypeptide chain A and B. They have linked by the help of this disulfide bond. Now this is able to control the blood sugar level. This we can say the mature insulin. It can able to control the blood sugar level. So this is done by the company Eli Lilly company from America. It's very helpful. Nowadays we can see millions of people say suffering from diabetes. So it is very helpful for these people. Those who are suffering from diabetics, it will be very useful. So that insulin we can say it is called humulin. It is called humulin. So what they have done? They have prepared insulin for the human beings from the human beings only. So that is called humulin. So this is one of the good application of biotechnology in medicine. Now we can study the another application, biotechnological application in medicine, that is gene therapy. Gene therapy. So let us see what is this gene therapy. You might have seen, you have heard the word like immunotherapy and chemotherapy. So there are radiotherapy, there are different therapies there. So one of the therapy here you can see gene therapy. What is gene therapy? It is used for the correction of genetic disorders. You may see, you have studied, you have observed, there are some people, means a small baby, they born with some genetic disorders. Yeah, some parents, they worry about that genetic disorders. How can we correct these genetic disorders? So we can correct, we can treat these genetic disorders by the help of biotechnology. That will we are going to study about the gene therapy. So we can study this gene therapy by the help of an example in the next session, in the next part. Please students, you can listen in the next part. Thank you students.